Howdy, today I'm Flippin Science looking at DNA replication. Thanks to Sam Moyle for producing this PowerPoint. The science understanding we're going to look at replication of DNA allows for genetic information to be inherited. Base pairing rules and the method of DNA replication are universal across all organisms. Describe the structural properties of DNA, including nucleotide composition and pairing, and the weak bonds, so those hydrogen bonds we talked about before. The importance of base pairing, AT and GC, and describe and represent the process of semi-conservative replication of DNA. So previously we talked about DNA structure, so sugar phosphate backbone, nitrogenous bases, A bonds with T, G bonds with C, and we talked about hydrogen bonds, which are joining those two strands together. So we get 2 hydrogen bonds for A and T, and 3 hydrogen bonds for G and C. This is called base pairing. So a adenine always bonds with a thymine with hydrogen bonds, and a cytosine always bonds with a guanine through hydrogen bonds. And this allows for low likelihood for error because it's very hard to have 2 bonds and 3 bonds combining in a different way. It doesn't really work very well. These hydrogen bonds are strong in terms of secondary interactions in chemistry. So any chemistry students, you'll learn about those, or would have learned about those, I should say. But they're also easily broken. So we can easily break this strand away from that strand, putting in a bit of energy and using some enzymes to do that. So we could use heat energy to do it, but we can also use enzymes, which are little proteins that do a particular job. So we can break these bonds apart and we can put in new ATs, Gs, and Cs on a separate strand and make 2 copies of what we started with. So we start with 1 copy, we get 2 copies, and we call that replication. So DNA replication, that's what we're looking at today. Summing up what I just said, DNA is made up of 2 strands of nucleotides, A bonds with T, G bonds with C. There's a wide variety of ATs, Gs, and Cs, and that's the genetic code, and they're joined by hydrogen bonds. An enzyme called DNA helicase unzips the double helix by breaking those hydrogen bonds between the nucleotides. Then free nucleotides come in and bond. So where you have an A that's exposed, a T comes in and bonds with it. Where you have a G exposed, a C comes in and bonds with it. Then another enzyme called DNA ligase. I think ligase, that means joining, ligature, for example. It comes in and joins the sugar phosphate backbones of the nucleotides together, and we end up with 2 sets of DNA from the one that we started with. And that's why it's replication. We're making a copy. Little animation showing it. So here's our enzyme. Our helicase is unzipping our DNA strand. We're getting our free nucleotides coming in, A's binding with T, G's bonding with C. Then we're having our enzyme, our ligase, coming in and joining the sugar phosphate backbones together. And once this is completed, we end up with 2 strands of DNA that are identical to the strand that we started with. Now you notice one strand kind of is copied in one direction, and the other strand, sections are done and then joined together, then sections are done, then joined together, and sections are done and joined together. That's just the way that the DNA gets read. It's 5' to 3'. You don't really need to know what that means, but it's important in terms of maybe future studies in biology. Here's another animation. This one shows a bit more detail. So here's our helicase. It's unwinding the DNA, the helical structure. Now the leading and a lagging strand, don't worry too much about that. We have DNA polymerase at the top, RNA polymerase will come down the bottom. You don't really need to worry too much about the difference between those two at a year 12 level. But you get free nucleotides coming in, the enzymes, the polymerases go along, and then we get the ligase joins them up. So that happens. It's not being shown in this animation, but the free nucleotides do get joined up. And that continues along the strand. So the DNA is unwound, free nucleotides come in, and they're joined together. And this is all moderated by enzymes. So here's another image. This one's static. So we've got a helicase, it's unwinding the DNA. Free nucleotides are coming in, and those nucleotides are being pulled in and put together by an enzyme called polymerase, DNA polymerase. And we've also got associated with that as a ligase that comes in and joins the sugar phosphate backbones together. We can see the direction of replication is a bit different for these. So one strand is going in this direction, the other strand is going in the other direction. Again, really at year 12 level, you don't really have to talk about that in any detail. So this method of replication leads to what we call semi-conservative replication. What that means is that each of the new strands of DNA that we get has one old half and one new half. So one strand is from the original molecule, and one is a new strand produced from that polymerase and ligase. So we get one old and one new, and that's what the semi means. So it's a semi-conserved, so it's half conserved. So you have half of your old bit, which is the conservative bit, and a half of the new bit. There's a crash course video that I'll link to on DayMap for you to watch that goes into this in a bit more detail. And so just summarizing that, so we start with our original molecule, and we end up with two daughter molecules. Each of those molecules is composed of an old strand and a new strand, and that's how we get our DNA replicated. So check. So replication DNA allows for genetic information to be inherited. Base pairing rules and method of DNA replication are universal. So describe the properties of DNA, including nucleotide composition. So we've got our nucleotide and our sugar backbone. ATGC, in the complementary base pairing, we've got our hydrogen bonds, which hold the two sides of the DNA molecule together, but they're fairly weak and they can be broken by enzymes or heat energy. And you need to talk about how semi-conservative replication works. So today on Flipping Science, we looked at DNA replication. That's it for Flipping Science today. See ya.